Hello friends, welcome to my channel Plant Science for You. Today in this video, we will discuss some molecular techniques which was preliminary used in genetics, like nucleic acid reassociation kinetics. In this, we will discuss genome complexity and curve specially. And in the second part, we will discuss physical mapping of chromosome, like heteroduplex mapping and restriction mapping. And in the later part of our video, and we will discuss more about molecular techniques. And so stay tuned and let's start the video. Let's understand what is the genome. The genome is the sum total of all genetic informations of an organism which stores biological informations, and it is basically a DNA or RNA. All eukaryotic and prokaryotic organism always have a DNA genome, and but in case of a virus, there may be either DNA or RNA, never both. And if you all know that the eukaryotic genome consists of two distinct parts, that is, one is the nuclear genome and another is the organelles, that is the mitochondria and also the chloroplastic DNA. Coming to the next slide, it is a very interesting term or concept, that is the C value, which is measured in picogram, and C value is the haploid DNA content, or the total amount of DNA present in a single set of its chromosome. Now, if we see what is the C value paradox, in general, if you can think that the organism who has the larger or greater C value or greater genome size, it must contain the greater genome complexity. You might thought, but it is not the case. There is always a genome size which is not equals to the genome, not implies the genome complexity. For example, the genome of a salamander contain more than 10 times of the amount of DNA that is in the human genome. Yet, salamanders are less complex than humans. You might think that salamanders have more than 10 times of DNA rather in case of human, but humans are more complex than the salamander. And this incompatibility, or you can say inconvenience, is termed as C value paradox. Next, what is the genome or sequence complexity? It is the number of times a particular DNA sequence, suppose GC, AT, TTC, this sequence is repeating or appears throughout the genome. That is the genome or sequence complexity. It is actually measured by renaturation kinetics, which was first developed by Roy Britton and David Kone in 1968. If we see any human genome or any other genome, there is the DNA sequence, and this DNA sequence possesses two kind of DNA. One is a highly repetitive or unique sequence, and another is a repetitive sequence. In the unique sequence, that means it is only one copy is present per genome, and we can find it in the prokaryotic DNA mainly. In case of repetitive DNA, it may be of two types, that is moderately repetitive, which is repeated from hundreds to thousands times. And in case of highly repetitive, it is repeated up to millions of copies. In case of moderately repetitive, it is found in mainly where the genes are regularly synthesized. That is, in case of rRNA synthesis, we actually need for our ribosome, and rRNA synthesis genes are moderately repetitive. Another is the histone protein synthesis genes, which are helping after DNA replication, the wrapping of DNA and forming the chromosome. And another is the transposable elements. And in case of highly repetitive, it is repeated up to millions of times, and it is actually the short tandem repeats. It is found, you can find it, it is in the centromeric region, the heterochromatic regions. And in the next slide, we have discussed about the nucleic acid renaturation kinetics. And renaturation means actually the reassociation, and we have utilized the complementary nature of nucleic acid. That means in the picture we can see there is a double stranded DNA, and if we denatured it utilizing the heat, and it will become the single stranded. And after that, if we cool or you just pull back the heat, and you will get the it renatured or double stranded DNA. And this renaturation kinetics or this renaturation process will give you two main information. There is the existence of repetitive DNA sequence, and another is the number of copies present in the genome. And how it is happen, let's see. That is, the highly repetitive DNA sequence will renature much faster, because many copies of the complementary sequence are present in the genome. That is why highly repetitive sequence or DNA sequence will renature much faster. And this renaturation actually occurs or happens due to random collision of the complementary strands, and so that the highly repetitive DNA sequences will be much in number in the genome, and they will get collision much number of times and will be renatured much faster. And this renaturation actually depends on the concentration of the DNA, the time given for the renaturation, and some buffer factor is also there, which actually helps in renature or in the renaturation. There is a viscosity factor, and in case of the pure DNA, there is no role of viscosity, but if there is a impurities like protein, there may be some viscosity factor also. This renaturation kinetics always follows the second order kinetics. That is, the rate of the renaturation will depend upon uh, the concentration of the DNA, its square. That is the second order kinetics. That is d c by dt equals to c square, is proportional to c square. Next slide. It is the COT curve. That is the C0T curve. COT means initial, C0 means initial DNA concentration, and T means time required for reassociation, and another is a buffer factor. We can multiply these three things and we get the COT value. Okay, its unit is moles per liter into second. It gives two very important information. Firstly, the complexity of the DNA, that is highly repetitive, moderately repetitive, or unique DNA. We get that percentage, that how much of the highly repetitive DNA present in the genome, how much unique DNA, and how much moderately repetitive DNA. And another information, that is the melting temperature of DNA. We can get from these values. If we see the curve, we can observe that if the G, suppose G or guanine is present in the genome for 20 times, then it will much faster reanneal or reassociate, because it will colloid, or it will two complementary G will colloid much faster than C, because C is present only 10 times. As G is present is in 20 times, it will much faster reanneal. And if we observe the curve very carefully, if we will see that in the y-axis there is a percentage of single stranded DNA. In the initial time, that is a very high when we start the renaturation process, and as the time progresses, the rapid rate of reassociation occurs in case of highly repetitive DNA. But then the intermediate rate occurs in case of moderately repetitive DNA, and in case of unique DNA, it is very slow. And this type of information we can get from the COT curve value. Next, our topic is the physical mapping of chromosome. In this slide, you can get an idea about physical map and linkage map. Physical map means we can differentiate or we can reassociate between two genes in terms of nucleic acid, or we can there may be some physical distance. We can distinguish those two genes in terms of physical distance, in terms of nucleic acid base pair. But in case of linkage map, we utilize recombination data and we get a relative location of two genes. Okay. So here the physical maps of chromosome depicts such recognition features such as sites for restriction enzymes, size of deletions, sites for AT-rich regions, etc. There are two main type of physical mapping is there. Firstly, heteroduplex mapping, and second is restriction mapping. We will see one by one. In case of heteroduplex, we have to know what is actually a heteroduplex. Heteroduplex means two strands of nucleotide which are not entirely complementary to each other. As because they are not entirely complementary, they are partially complementary, and that place where they are not complementary to each other, that will form a loop, and that is the heteroduplex. Suppose we can detect any deletion mutation by comparing the heteroduplex. For example, in a DNA duplex, one strand may contain one allele for the gene, and while the other strand may have another allele or the mutation is there. And that because of that, their sequence, DNA sequence will not match or not be complementary, and for that there may be a formation of loop, and that will help us to mapping a deletion or duplication in the heteroduplex or DNA sequence. Here there is a given is an example such as heteroduplex molecules are prepared by mixing two denatured DNA preparation. One is a wild type and another is a mutated one, and then they goes for renaturation, and we get a loop in the wild strand, as there is a in the mutated strand there is a missing or deletion there, and we get a loop in case of when a wild standard DNA, and this form a heteroduplex, and we can compare this to get a deletion in the particular location. In the next slide, it is a restriction mapping. This restriction mapping, we use the restriction endonuclease enzymes to map a particular vector or a circular DNA, and it depicts the site where one or more in restriction endonucleases produce cut in the chromosome. Its several approaches are used for restriction mapping. That is, example, comparison of products of partial digestion with enzymes and sequential digestions with two or more enzymes, different restriction enzymes. We can make this restriction mapping. In this case, if we in the lowermost picture, we can see that there is a circular DNA and it has three restriction enzyme site. And if one enzyme, if that particular restriction enzyme is given in the medium and the complete digestion is done, then three fragment of DNA will be produced. One is of 3 kb, another is of 2 kb, and another is of 1 kb. And if we run the gel in the gel electrophoresis, we will get three clear cut band, and we can say from that there is a complete digestion, and we can find out the restriction site from this by mapping. Next slide. Here there is a partial diagram or partial digestion of circular DNA. If there is not a complete digestion, there may be some partial digestion, that is the enzyme cut one restriction site and miss one and cut the another site, and that will produce different lengths of DNA. You can see the consequences. In the first case, if we see there is a circular DNA, there is no cut is produced. In the second case, there is a cut is at only one place, that will produce a linear DNA fragment. In the third case, there is a two cut is present, and there is the two band will be produced. In the fourth case, there is a three cut, and there will be produced three fragment. In case of fourth cat, fourth case, there is a complete digestion. That is, there will be always a complete digestion in case of partial digestion, and that will produce four DNA fragment. And by comparing these fragments, we can derive a restriction map of any vector DNA, circular DNA. So this is up to this visual lecture comes to an end, and in the thank you for watching. And in the next video we will discuss about nucleic acid hybridization probes, southern blotting techniques, northern blotting, western blotting, etc. So thanks for watching. Please subscribe the channel and share.